associated with this Science Dialogue series, and she's assistant professor at KGPU,
APG.
University Baroda. Now, madam will continue.
Okay, thank you so much, sir. Thank you, Gopalji, sir. And first of all, I would like to welcome Ravi, sir, and all our participants for this lecture series, which is known as Science Dialogue series. This is an initiative for the scientific community or for the research student, for the undergraduate, postgraduate, whosoever is willing to learn from the pioneers or the peers from the bioscience. They can enroll and they can get the knowledge about any topic which we are discussing in this Science Dialogue series free of cost. This is a complete free of cost series. So this is just to be a make you all aware that why this Science Dialogue series is here. Now it's time to introduce everyone with our today's speaker, Professor Ravi Vijayvargia, sir. Sir, first of all, welcome to you for this series, for today's session. So regarding his introduction, sir is an assistant professor in Department of Biochemistry, the Maharaja Sayaji Rao University of Baroda. Apart from this, he is an assistant director at the CMB, which is Cell and Molecular Biology Department of the MS University of Baroda. Regarding sir, regarding his research or regarding his all the credentials, if we talk about, sir is a very enthusiastic scientist. He is a very enthusiastic academician, as it is being very obvious from whatever is available or whatever we are knowing about him. Sir has guided many students through their research project in their MSc dissertation, in their as their PhD guide. He is guiding so many students. He has completed few of the research project from the GSBTM, from the DST SERB or ICMR. He is also working currently on the neurodegenerative disorder, among which one of the Huntington disease, which is very known to anyone who is related to prion biology or who is related to the diseases which are because of the malfunction of the protein. So his interest lies in that, and apart from this research curriculum or whatever he is doing at the academics, he has been very instrumental in guiding the student apart from the regular practice. He has been a resource person in the various FDPs or various
आपकी आवाज नहीं आ रही है या मुझे नहीं आ रही आवाज?
So
हेलो.
this is just a very brief introduction, and he has been awarded by different agencies. One of them is from the medicine department of New Jersey, where he has been awarded as a Gallo Award for his research in this field. So, sir, this is very brief introduction, and apart from that, what I can say, sir, I myself has a little bit interaction with you while we were preparing one abstract book for the MSU National Science Day in 2021. So in that I was in the committee with you. Under your guidance, we have prepared one abstract book for the National Science Day. So Gopalji sir, I am also, maybe sir is not recalling or he may not be remembering us, but that was my first interaction with sir for me, and it was very useful for me because he has guided us how to just make those abstract book, aligning them, and how the according to the theme how we can adjust that. So that was my first introduction with the sir, where I was actually guided by him. So sir, it was my pleasure to have the interaction, to be working with you. And now again, since I am introducing you, so this is again a pleasure for me to introduce to our today's participants, and I hope they may be getting benefited from the lecture that you are going to deliver. So, sir, now the this platform is yours. The online virtual platform is yours, and we hope that our participant may be interacting. They may be asking their queries regarding what we are going to learn from you today. So, sir, please, uh,
Yes.
you can start your session. You can start your lecture.
Sir is
Gopal.
also resource person in all my online courses in
Yes.
recombinant DNA technology and genomics and proteomics. And
Yes.
most of the
Yes.
time in that also he has never asked his on radium and means there is without conditional. We have also spent 2 years with him at
Yes.
MS University, so I know he's very down to earth. And people claim to be a professor in the this 20 years of post PhD experience. He has 20 years of post PhD experience,
Yes.
including 10 years in top institutions of USA. Sometimes in
Yes.
government institution, people think ki whether designation is a professor or dean or director or not. But I don't consider that designation. I know his credential, and he has more than 20 years of experience in which people claim to
Yes.
be a professor and dean and director. So I request all the participants to interact with him. You can extract as maximum as possible from him. He will not stop you, and he is not like a lecture that you can only ask after completion of the lecture. You can raise your hand and you can ask in between also, because ये याद करके नहीं lecture दे रहे. Whatever he is delivering, he has experienced, he has performed himself. On Huntington disease, he has worked in postdoc also, in assistant professorship also. 11 years of assistant professorship he has spent at MS University, and before that 10 year, more than 10 year at USA. So I request you all to interact, concentrate on the lecture. Just don't join only for certificate. I always repeat this thing, है ना? So yes, sir. Please continue.
Thank you, Dr. Gopal ji, Dr. Poonam, for kind introduction. And as I can see now, around 22 participants are there. So, so it will be interactive. Okay. So whenever you feel like you have any, am I audible clearly?
थोड़ा कम हो
Yes.
जा रहा
Yes.
कभी कभी.
Okay. Because right now the Wi-Fi in the lab is not working, so I'm using my hotspot. So that is
अच्छा, मैं भी हॉटस्पॉट ही कर रहा हूँ.
So that is potentially the reason. Okay. So if the Wi-Fi comes, then it will be better. But right now it is on hotspot. So, but it is audible, no?
Yes, yes, audible.
Yes, sir.
Okay. Okay. So as Dr. Gopalji said that today I will be sharing my research experience, which we have done from last almost 20 years of research on one particular neurodegenerative disease, which is called Huntington's disease. So, so it will be interactive. So whenever I ask, you can raise your hand or you can unmute yourself and answer. Okay. So that I know that, because once I start sharing the slide, I'll not be able to see your raise hands also. I have to again go back and see. So you can unmute yourself and ask. Okay, whenever you have any doubt or any query, unmute yourself and ask me. Fine with everybody? And should we use pure English or mix of English Hindi? What kind of method or you'll prefer? English, okay. Khushi is saying English, okay. So it's fine, okay. So we'll be using both languages, mix of both languages. Some technical terms I have to tell in English, otherwise Hindi maybe I will try to explain. Fine. So, how many of you have heard of this disease? कितने ने सुना है Huntington disease सुना है? Yeah, one or two can unmute and say, or yeah, okay. So you have heard of this disease, okay? So it is a neurodegenerative disease, as the name suggests. It affects the nervous system, okay, brain or spinal cord and other associated regions of the brain, okay. So the more common disease which you must be aware of, like Alzheimer disease or Parkinson disease, they are more common. Okay, but Huntington disease is also not very rare. It is a rare disease, but not very rare. But the main thing is there is no cure. Okay, so Huntington disease is as of now an incurable neurodegenerative disorder. Okay, so today what we are going to see in this 1 hour or so, that what causes the disease, and based on the title, okay, if you have seen the title of the talk, so do you remember what is the title of the talk? Anybody? Nobody remembers the title of today's talk? Okay, it says Huntington disease, a tale of amino acid turning deadly, isn't it? So here we will see a case in which how a normal amino acid can become toxic, okay, and that can cause the disease. So I'll start sharing my slides, and as I said, whenever you have any query, you can unmute yourself and ask me, okay. And in between I'll keep coming out of the presentation and check the message boxes, so that also, but not frequently, so that there is not much interruption, okay.
मैं
So
बताऊंगा आपको चैट बॉक्स में से. If they are interested, they can unmute and ask, है ना?
yes, okay,
Yes.
okay. So First, even before I start, I see one question. Anshu Kumar is asking, what differentiates the disease from Parkinson's and Alzheimer? Uh, the causative, okay, all are neurodegenerative disorders, but the cause, okay, Huntington disease is pure genetic disorder, okay. It is caused because of a mutation in Huntington gene. While Alzheimer, only 30% of Alzheimer patients have genetic cause, 70% are non-genetic. Okay, so although sometimes the symptoms can overlap, like for example, Alzheimer, you have a memory problem, memory loss. That memory loss also is seen in Huntington disease, because the brain regions where memory is stored in several regions. But if the disease is affecting those regions of the brain, you can have overlapping symptoms. Okay, तो वहां पे अल्जाइमर और पार्किंसन और हंटिंगटन के सिम्टम्स भी मैच करते हैं. फॉर एग्जांपल पार्किंसन में if you have a motor control is lost, same thing happens in Huntington's also, because the same regions like substantia nigra, okay, striatal neurons are killed in both of them. Okay, so whenever there is a common region of brain affected in a disease, you can have overlapping symptom. But Huntington disease is a pure genetic disorder. Okay, so it is can only be caused because of a mutation in a gene called Huntington. Okay, so let's see what exactly mutation is causing and how the title justifies it. Okay, so let me start by sharing the slides now. Okay, can you see the title slide? Is it
Yes,
visible,
visible.
Yes, sir.
title slide?
Yes, sir.
Okay.
Yes, sir. We can see.
Okay. Yeah. So you can see that it reads that on purpose, I made a little change in the title. You can see Huntington's disease, a tail and in bracket tail of amino acid turning deadly. So you soon know that why I have written other tail in bracket. Okay. You know the difference between tail and tail?
Yes, sir.
What is the first tail
Story.
and the
Kind of
second tail?
story.
Second tail.
Dog's tail.
Tail is a
Okay, so here
follow-up.
also there is tail is there, but also tail is there. Okay, so that's why I should have kept this title from beginning, but later on I realized that that is today at least I can put it that way. Okay, so here we'll see what is the tail and what is the tail. Okay, so as you know, for any disease or anything, first we should know the history. So first term, this Huntington's disease, actually originally it was called Huntington's chorea. I don't know whether anybody have heard of this Huntington's chorea term, but initial term was coined by a doctor called George Huntington way back in 1872. Okay, that time it is called Huntington's chorea because the main symptoms associated with Huntington's chorea was like dancing like movement. Choreography. Choreography. सुना है सबने word choreography?
Yes, sir.
Yes,
Okay,
sir.
choreography is you dance के लिए ना. So the Huntington patients, they because of the uncontrolled movement, when they used to move, it used to look like, or it looks like that they are dancing. Okay, and that's why the word chorea was given, Huntington's chorea, because of the dance like movements. Okay, but later on when the patients were associated with other symptoms like memory loss or psychological problems, then it was termed as Huntington's disease. Okay, so from chorea it was named as disease. Okay, so the first uh, terminology was given by George Huntington in 1872. And what exactly causes it? So here it is. So also this gene, I told you, Huntington TIN, Huntington gene is present in all animals. Okay, invertebrates, what all vertebrates, sorry, all vertebrates it is present. And what exactly cause a disease, that I'll use the pointer so that you should be able to also. Okay, so you can see here, in case of human, the gene is present on short arm of chromosome number 4, and it is a big gene which has almost 63 exons. You can see there are different exons. All of you know the difference between exon and intron. Intron
Yes,
और
sir.
exon का difference पता है ना?
Yes, sir.
So it is
Yes.
a
Yes,
big
sir. Yes,
gene,
sir.
has 63 exons, which eventually after splicing come together to form the Huntington protein. What is peculiar about this is, in exon 1 of this gene, there is a repeat of a particular codon called CAG. You can see CAG here, है ना, CAG
Yes,
दिख रहा
sir.
है?
Yes, sir.
हाँ.
Yes, sir.
So
Three
in
time,
exon 1
three
there
time
is a
repeated.
repeat of CAG, and this CAG repetition uh, is polymorphic in the sense different people can have different number of CAG in Huntington gene. So in human population you can see different people carrying between 10 to 25 repeats. Okay, so this CAG is a codon for a amino acid called glutamine. Glutamine सुना है सबने?
Yes, sir.
Yes, sir.
Yes, sir.
So when there is a poly CAG repeat in the gene, it results in a polyglutamine repeat in the protein. So ये नीचे जो है वो protein है, okay? So this CAG repeat in the gene has resulted in a polyglutamine tail. Okay, that's why the tail word was used. So a polyglutamine tail is formed in the protein. Okay, so this is the normal Huntington. Okay, normal gene with a CAG repeat between 10 to 25 is the most common found in the human population, and it translates into a polyglutamine tract or tail in the protein. Okay, and this is vital for normal function in neuronal cells. If this is present, it will have a normal function and neurons will be happy. Okay, but there are mutations in which the CAG repeat can expand. Okay, so when CAG repeat expands and the repeat size increases and it comes between 40 to 80. Okay, from between 10 to 25 is actually basically speaking 10 to 35 is normal, 36 to 39 is intermediate, and 40 onwards it becomes pathogenic. Okay, so when there is an expansion of CAG, and that happens because of the slippage of DNA polymerase during replication. Okay, so the DNA polymerase during replication sometimes it slips and it goes back and bind upstream, and that way it ends up adding more CAG. Okay, so because of slippage of DNA polymerase and if the event is not corrected, it leads to additional CAG and the repeat size can increase. Okay, so if repeat size becomes higher than 39, that is 40 onwards, now what happens that this protein, although nothing else has changed, only the CAG repeat has resulted, increased CAG repeat has resulted in additional, the length of or the tail of polyglutamine has increased. Okay, and this addition of additional glutamines can alter the structure of the protein, and when the structure is changed, you know, the function can also change. Okay, and now it becomes a mutant protein, and this additional glutamines can lead to misfolding of the protein, aggregation of the protein, and all the normal functions which it carries are no more carried by this, and that's why when there is this, you can see this leads to accumulation in the cell, aggregation in the cell, and when the functions are not carried, eventually the neurons, they are killed. Okay, they die. Okay, and that's why whichever area of the brain the neurons are getting killed, the corresponding function which they carry out will be affected. So you see a memory loss, or the neurons which control your bodily movements, you will see a change in bodily movements. Okay, so this is how the disease occurs, by expansion of CAG, expansion of polyglutamine tract in the protein, misfolding of the protein, aggregation of the protein, and thereby the cells are getting killed. Okay, so what all pathologies, because of the aggregation of the proteins, what all pathways are affected, we have studied over the several years. Also in my postdoc we studied that how this expansion alters the protein structure. All those stories or all those tales I am going to tell you today. Okay, so whenever you have any doubt, you ask me. Okay, interrupt me and ask me. अभी तक सब clear है?
Yes, sir. Yes, sir.
Clear till now?
Yes,
Yes,
sir.
sir.
Yes, sir.
Okay.
Yes, sir.
Okay, so as I said earlier, this is a totally genetic disorder. Okay, so if there is even one parent is affected, for example, in this case, affected father means one of the allele, chromosome 4 allele, has a mutation, while mother is fine. Okay, then also 50% of the offspring, even because this is a autosomal dominant disorder, that even if you have one allele which is mutant. The second allele is normal. Blue is normal allele. This is a mutant allele. Even if there is a second allele, a normal allele, then also the person is going to have the disease. Okay, so you can see if only one parent is affected, 50% of the next generation will be affected by the disease. Okay, so this is how it is passed on, and it is totally genetic. Also, as I was just saying that in in a given population, people can have different CAG repeats. Okay, so 10 to 26 they have is a totally normal repeat size. If they have between 27 to 35, it is called intermediate size. If there's between 36 to 39, it is called reduced penetrance, means some people might get disease, some might not, so reduced penetrance. But if it is 40 and above, it becomes fully penetrant. That means everybody, each and everybody who has 40 or more repeats is bound to get the Huntington's disease. Okay, and there is an inverse correlation. Okay, so you can see here, this is based on the population studies. On the x-axis you can see the CAG repeat size. On the y-axis you can see age of onset. That means at what age the patient will show symptom. Okay, so it can be motor symptom where the body control is lost, or it can be memory problem. Whatever symptom is shown, that is called age of onset. Okay, so you can see as the CAG repeat increases, the age of onset decreases. It is in years. Okay, you can see in years. So as the CAG repeat is increasing, there is a decrease in age of onset. Is it clear to everybody? दिख रहा है?
Yes, sir.
किसी
Yes,
का
sir.
किसी का CAG repeat अगर 40 है, तो उसका age of onset average. This is line is the average. Okay, and the shaded blue part is the standard deviation. Okay, so you can see the average. If the CAG repeat is around 40, average age of onset will be around 65. Okay, here you can see around 65. Okay, but what you can see that this is a standard deviation is huge. So if you consider standard deviation, people having 40 CAG repeat, some people can be protected till age of 78, but some unlucky people can get the symptom as early as 27, 28 years. Can you see this difference?
Yes, sir.
So this is what is called a modifier effect. Okay, that your disease is going to be there once you cross the CAG repeat of 40. But when the disease will come, there is a big, like almost 10 to 20 years differences between people. Okay, and that is called modifier effect. Means modifiers can be your genetic makeup, what kind of food you eat, what kind of environment you are in. All those can affect that whether you will get an early onset or a late onset. Okay, so since I said there is no cure for the disease, so there are two ways in which this researchers target this disease to find a cure. Directly, one is reducing the CAG repeat to make the wild type protein into normal protein, or sorry, mutant protein into wild type protein, or totally eliminating the mutant allele. म्यूटेंट allele को हटा ही दो, तो wild type allele जो second है, so that wild type allele should be able to carry the normal function. Okay, okay. So as I said, the CAG repeat has inverse correlation with age of
Shubham
onset.
has asked one question.
Yes,
Huntington's and spirocerebral ataxia both have CAG repeat expansion. What is the difference?
yes. Both are CAG repeat disorders, but the genes are different. Okay, ataxia have ataxin gene in which the CAG expansion happens. In Huntington's disease, Huntington gene has CAG expansion. So both are different genes in which expansion is happening. Clear?
Okay, sir. Thank you.
Number of CAG repeats and disease severity also have a direct correlation. More the CAG repeat, more severe the disease will be. Okay, so these two correlations are there. So what are the symptoms? So as I said, initially it was called Huntington's chorea because of the motor symptoms, but later on several other features were associated. So you can have cognitive decline, that is memory loss. You have mood swings, like you can have like similar to what happens, like in bipolar disorders, that also can happen. Then involuntary movements, the motor control is lost. Then hallucinations can happen. speech difficulties can happen, or also behavioral or personality changes. So there can be multiple features, but not all patients might get all the symptoms. It depends on what kind of which brain area is affected more. Accordingly, symptoms can come, but different patient can have different symptoms or mix of symptoms. Okay, so as I said, there is right now no cure. So only the treatment is focusing on the management of symptoms. Okay, so based on the symptoms, the drugs are given to patients. So what are the functions? Because when the mutation happens, so many things are dysregulated. So what are the normal functions of the wild type Huntington protein? So these are the normal functions of wild type Huntington protein. It helps in transcription, in vesicular trafficking in neurons. It has anti-apoptotic activity. It is also helpful in energy metabolism, coordination of cell division, also the transport. Okay, so there are multiple functions associated with wild type Huntington, and when this mutation happens, the wild type protein becomes misfolded, and the also this protein is a huge protein. It is a 350 kilodalton protein, and there is no enzyme activity associated with the protein. So main function of this protein is to act as a scaffold. Okay, because it's a huge protein, it allows many molecular interactions, and it is a kind of site for so many other enzyme activity. It doesn't have activity of its own, but it can act as a facilitator of other for other complexes to come dock to this protein and do their activities. Okay, so what exactly here you can see, when there is a normal Huntington, you have a reduced polyglutamine length, and when it is mutant, there is an expansion of the glutamine chain, and this leads to misfolding of the protein. It There is loss of wild type function, and then toxic gain of mutant function, because when the protein gets misfolded, it can, because of the change in properties, it can have new functions. Okay, that is called gain of function. So also misfolding also exposes some of the protease cleavage site, and the misfolded protein can be further cleaved into smaller fragments and the N terminal fragments, because the CAG repeat is in the 5' end of the gene. So the polyglutamine repeat is also on the N terminal of the protein. So you get a small N terminal fragments, which is again they are small, so they can migrate to nucleus, and they can cause problem in nucleus also by aggregating there. So you can have toxic oligomers, and eventually because of the oligomer formation and interaction with several proteins, non-specific interaction with several proteins in the nucleus, then you can have you can have disarray of the transcriptional machinery. And if the transcriptional machinery is sequestered by this mutant protein, there can be a lot of transcriptional dysregulation. A lot of genes can be downregulated, upregulated, and that can lead to toxicity as well as neuronal cell death. Okay, and this is only one path. Okay, as I'll show you in the next slides, there are several other pathways which get affected by the mutant Huntington protein. Okay, so here you can see that
दो
one.
क्वेश्चंस हैं.
हाँ, पूछिए.
एक अंशु का है तो सजेशन दे रहे हैं, सर टीआरएनए मॉडिफिकेशन इन कोडिंग फॉर ग्लूटामिन कैन इट बी इंक्लूडेड इन हंटिंगटन क्योर? टीआरएनए मॉडिफिकेशन इन कोडिंग फॉर ग्लूटामिन, तो पूरा ग्लूटामिन का चेंज हो जाएगा भाई, ऐसा थोड़े होगा. ग्लूटामिन का टीआरएनए चेंज क्या बोल रहे हैं? कैन यू अनम्यूट एंड स्पीक? अंशु बोलो. ओके, इज नॉट हियर. सृष्टि का क्वेश्चन है.
No, sir.
हाँ अंशु बोलो. यस अंशु.
Sir, actually, I wanted to ask that if we do some modification, like reversing the anticodon and codon interaction part of tRNA, so then can it be included in the cure of Huntington?
So you are trying to say that the tRNA should not recognize the CAG codon in mRNA, and that why not add the glutamine. That's what you are saying?
No, sir. Actually, if we do the specific recognition.
No. See, normally what tRNA will read your mRNA, whatever codons are there, the anticodon loop of tRNA will read that and add the corresponding amino acid. Okay,
Yes, sir.
so in that sense,
Okay, so
what
will it
I thought
affect
what you
all
are
the
proposing is that the tRNA, if it is not able to read the uh, codon for glutamine, it will not be able to read the, it will not be able to add glutamine, and then the translation will stop. That's what you are trying to say.
yes, sir. Like if we do
Yeah.
some nonsense,
So
nonsense.
the biggest problem with Huntington disease is that even the wild type has glutamine chain. Okay, wild type also till 35 repeat 35 glutamine is wild type. So
Yes,
the differentiator is not a mutation which like one amino acid has changed from glutamine to aspartate or glutamine to arginine. It is amino acid is same glutamine only, the number is changed. Okay, so that's why even targeting CRISPR Cas9 or any other gene targeting also becomes difficult, because this uh, therapies can target the wild type allele also, because wild type also has CAG repeat till 35, it is normal. But if you are targeting a CAG repeat, even the wild type allele gets targeted. That is the biggest problem. Getting the point?
okay, sir. Sir, then my question is, what causes these repeats to increase, to expand in the, particularly in
That's what I said
Huntington's? What is
during.
the, what's the
So that what it is a during the replication event, the DNA polymerase, when it is trying to copy the Huntington gene, when repeat size are on the higher side, means like 25 plus or so. Okay, so what happens, because of the multiple repeats coming, there is a tendency of DNA polymerase to slip. Okay, slip means it falls off from the template strand. Okay, and when it binds back, sometimes it binds upstream from where it fell down. Okay, so if binds upstream, it ends up adding additional CAG, and in the next round, because of the additional CAG, the corresponding strand will add up their the other bases, the corresponding bases, and it ends up having more CAG. Okay, because normally it gets corrected, but if it is missed, then you can have more CAG in the daughter cells. And when it happens in the germline cells, it can pass on to the next generation.
Same question asked by Srishti Agrawal. Srishti, you got answer?
Yes, sir. Yes, sir.
Yes.
Yeah.
And Mohammad Bakir, you have just joined and you are asking feedback per forma. Please wait. And we are not conducting all these things to give certificate. Yes, you will attend, then you will get. But only just you have joined. I have seen. I have allowed you. You are asking feedback per forma. I will share. So be patient, Hannah. Keep patience. Yes, please continue, sir.
Okay. So as you can see here, you can see that mutant Huntington, it has tendency to oligomerize and aggregate, and when it does so, it also interacts with different organelles like mitochondria, or it can also cause glutamate excitotoxicity. Also, because of the misfolding, it can cause ER stress, oxidative stress, and also axonal transport, which is has normal function is axonal transport will get impaired because the mutant protein is aggregated. It cannot carry the axonal transport. Autophagy also is affected because of the large size of aggregates. So all also when it goes, the smaller fragment goes to nucleus, the nuclear translocation, it can uh, form inclusions with other proteins in the other transcription factors, and then can lead to dysregulated transcription. So based on multiple dysregulated pathway, eventually what happens that the neuronal cells where these all pathways are affected, eventually it's become non-functional and it degenerates the neuron. So whenever the neurons are degenerated, what you see in this is actual patient sample. So you can see in normal people, this area you can see here versus in patients. So this is the white area is because of the loss of neurons. So in the lateral ventricles, you have a lot of neurons lost, and eventually other areas. So the first target, the basal ganglia, lateral ventricles, these are the first target of the mutant protein, and eventually cortical regions all are affected. Okay, and you can see the because of the loss of neurons, you see the empty spaces here. Okay. So as I said, there is no cure, but since there is no cure, it doesn't mean that we cannot or we should not research. So there are different trials also with different levels. Some trials have successful trials have held till stage 2, some are have reached stage 3 also, but there is no cure as such. Okay, so there are two main ways in which people are trying to address this. One is at gene level, so targeting the mutant Huntington allele. So you know that first simple way was to target the CAG expansion. So if you are able to target the CAG expansion, reduce it, then if the mutant allele can become wild type, and but it's difficult because it is also targeting the proteins which are very costly, because they have to be injected directly into brain. Although they have shown in clinical trials to lower the mutant allele, mutant protein, but to come into real practice they will be very difficult because very costly. Second kind of targeting is to target at the protein level. So the protein misfolds, and because of the misfolding or aggregation, several pathways are affected. So if you can prevent the misfolding of protein in some way, or if you can, the pathways which are affected, if you can bring them or normalize them, potentially the neuronal cells will not die. Okay, so there are several small molecule candidates which have been uh, in clinical trials on basic research, which are targeting the protein at the protein level, also targeting the post translational modification of the Huntington protein. Do you know what is post translational modification? Can you name some post translational modification of proteins?
Methylation, acetylation,
Glucose
glycosylation.
Yeah. Okay.
Phosphorylation.
Phosphorylation. Very good. Okay. So what it is seen that there are several modifications which are different in wild type and the mutant protein, because mutant protein has misfolded. Okay. So misfolding of the protein can bring some of the residues which were buried in the wild type onto the surface, and vice versa. Okay. So the PTM pattern of the wild type and mutant are different. So you can target the modifications, and that targeting of some of the modification can reduce the toxicity of the mutant protein. Okay, so these are different ways in which you can potentially find a cure to the protein. Also, the third thing which I have not listed here is, I that is not cure, but at least you can delay the onset of disease by targeting. If you are first identifying and targeting the modifiers, I showed you na that person having the same CAG repeat for example 40. Some person can have the disease, average was around 65 years of age, but somebody can get at 85 and somebody can get at 25. तो याद है ना वो ग्राफ दिखाया था.
Yes, sir.
Yes, sir.
So one way is to identify what modifiers are there in that person, and if you can target that. Okay, so if somebody is able to live symptom free, अगर 85 ईयर में symptom आता है तो उस person ने almost normal life जी ली. You agree or not?
Yes,
Yes,
sir.
sir.
So if you are able to delay by 10, 15 years age of onset, basically the person having the normal life. So that is another way people are targeting, that you identify the modifiers, and if they, like for example, easiest to work on is changing the dietary habits, changing, sometimes you can, if you want, the environment is very polluted and you can move to a different place if possible, so you can change the uh, effect of pollution or the environment also. Okay, so these are doable things if that you can do change in the diet or environment, and if we can prevent the disease onset by 10 years or 15 years, it is almost good as getting a cure. Okay, so that is another arm of research people are doing. Okay. Okay, so now starting with my research. So first I'll show you some data from my postdoctoral research which we carried out at Harvard Medical School and MGH in Boston, and then I'll show what we are currently doing. Okay. So till now everything is clear to everybody. अभी तक सबको जो बताया समझ में आया है?
Yes,
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 sir.
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 Yes, sir.
Yes, sir.
Okay. So now, yeah, now I gave you background. Now we will.
Sir, just a minute.
Yes.
I have one query, sir. This Huntington disease, is it involved in the genetic test that is done during the trimesters or the for the
No, no,
prenatal stage?
it's not,
Is it
it is not involved. It is not, because it is a very rare disease. Okay, that's why it is not like,
in
not like
India the
having Down syndrome.
prevalence? Yeah, prevalence of Huntington is not so much in India. or
because it is because of the lack of the large scale population studies, but almost it will be similar to European population. But since there are no large scale studies done in India, we don't know exact prevalence, but it is still around 4 to 5 people per 10,000.
okay, sir.
Okay. Okay, so this is a prediction based on the sequence of amino acid. We made a prediction that what kind of secondary structure Huntington protein can have. So as you can see here, uh, in red is the alpha like, green, yellow is the random coil, and gray is the beta sheet. So basically, most of the protein you see it is in red. Okay, so and those red regions are also they are marked together. So you can see those regions are again in the second uh, this tick model. You can see they are in the cyan color. So they are called heat repeats. Okay. So whenever these are alpha helical repeats which come together, they are called heat repeats. And this is the polyglutamine. I told you the polyglutamine tract is in end terminal of the protein. So here is the polyglutamine tract, and this is the based on the just the prediction. Based on the prediction, we can see that this protein is basically a alpha helical protein, like secondary structure of the protein is an alpha halide. Okay, but we wanted to confirm that whatever we predict, is it true or not. Okay, to confirm we have to do actual experiment. Okay, for that we need actual protein. So our lab was the first lab in the world to be able to purify both wild type as well as mutant Huntington, like to almost 100% purity, and that we can then use for different biophysical techniques to find the secondary structure and tertiary structure. Okay, so here you can see this, we used a insect cell baculovirus system to express and then use the affinity purification to purify the protein. So here you can see a series of Huntington proteins, full length human Huntington proteins with different glutamine chain repeats Q2, 23, 46, 67, and 78. So 46, 67, and 78 are in the mutant range. Q2 and 23 are in the wild type range. So we could, so you can see this is a huge protein. This is a molecular weight marker in kilodaltons. So this is 250 kilodalton, but our protein is 350 kilodalton. So this is a 250 kilodalton protein. So you can see we could purify all the proteins, and as I said, this was way back in 2010 or 11, but that time we were the only lab in the world to be able to purify the full length protein. Okay, so since we could purify it, we confirmed by Western blotting also using the specific antibodies, and then we went to do different assays to determine its secondary and tertiary structures. So you can see this is data from circular dichorism. Have you heard of this term, circular dichorism?
Yes, sir.
Yeah, so
No,
this is
sir.
a common technique in which we are able to determine the secondary structures. Okay, so in this case protein. Okay, so you don't have to go into details of it, but basically there is already established uh, kind of what kind of, if there is alpha helical protein, what kind of this is a wavelength scan. Okay, so the different wavelength in nanometer you can see on the x-axis. So a wavelength scan is done using different wavelengths. Here you can see from 190 nanometer to 260 nanometer. So you do a wavelength scan, and based on at what wavelength the maximum absorption is there and minimum. So there should be a maxima and a minima. So in case of alpha helical proteins, you see a maxima at 192 nanometer. So you can see a maxima here, and there is a two minima at 208 and 222 nanometer. Okay, so you can see two minimas. So all, whether it is a wild type protein or mutant protein, you see the maxima and minima. Okay, but if you quantify them, you can see this is a quantified graph, that as the polyglutamine repeat length increases, there is an increase in the alpha helicity. Means the alpha helical structure, the structure become more helical as the glutamine repeat increases. Okay, so this was about the secondary structure. But what about the tertiary structure? Okay, to determine the tertiary structure, normally what we do, we make a crystal. We make a crystal of the protein, and you can do a x-ray refraction, and or from using the crystal you can determine the 3D structure of the protein. सुना है सबने x-ray refraction?
Yes.
Okay, So in case of Huntington, the problem is this protein is has tendency to oligomerize, है ना? और खासकर purified form में when we have large amount of it even the wild type protein have tendency to aggregate. Okay, so it is very difficult to get in monomer form. So to get the monomeric form of the protein, we have to do a sucrose density gradient, and as you can see here, this is the input. So you can see multiple bands are seen. This is a blue native page. Have you heard of native page?
Yes, sir.
SDS page and native page. So this is a blue native page. So as you can see in the input, there are multimers. Okay, these are oligomers of Huntington. But when we run on sucrose density gradient, based on the density of different oligomers, they come out in different fraction based on the gradient. Okay, and you can see in the fraction 7 and 8, we can get a monomeric fraction. Okay, so similarly, for this is for wild type protein, this is for mutant protein, we got a monomeric fraction, and these monomeric fractions then we subjected to. So this protein is the monomers are also very not, you can say, heterozygous. Okay, not or so they are not having homogeneous structure, so we cannot, they are not, we are not able to make crystal out of it. Okay, so if you are not able to make crystal out of it, then you are limited by solving the structure. So but fortunately at that point of time, new technique had come, which proteins which don't form crystals used to determine the your tertiary or quaternary structure or crystal structure, what you call. So that, do you know what technique is that? Have you heard of cryo EM?
Yes, sir.
Yes, sir.
क्रायो ईएम बनाने वालों को नोबेल प्राइज मिला रिसेंटली, पता है ना? So cryo EM is the technique which we used. So cryo, what cryo means का क्या मतलब है? Cryo, what it
So
cryo
cold,
का मतलब क्या होता है?
cold,
क्रायोजेनिक इंजन, है ना? क्रायोजेनिक
cold,
इंजन,
sir.
तो हाँ,
Cold crystals.
very cold. Okay, cryo means very cold. Okay, so first you have to freeze down. Okay, you freeze the monomers into crystal. Okay, इतना ठंडा टेम्परेचर लिक्विड नाइट्रोजन में डुबो दिया. Okay, so you freeze them so that they cannot change their, they are frozen in whatever conformation they have. So you freeze them in whatever conformation they have, and then you scan. So cryo EM में you freeze first and then scan it, and then using the software you can determine the structure. So that is what we did. So you can see here, uh, in the bottom you can see the actual cryo EM image, and using the software you can determine how exactly it will be. Okay, also we used the cryo, this is the wild type protein, and then this wild type protein bound with a specific antibody, so you can see where exactly the antibody binds, and this is a mutant protein. Okay, so this is a mutant protein alone, this is mutant protein bound to the antibody. Okay, and you can see here between the wild type protein and the mutant protein a difference in structure, isn't it? दिख रहा है?
Yes.
Okay, so because of the just expansion of polyglutamine chain, the monomeric structure of the wild type and the mutant protein gets altered, and that alteration in the mutant Huntington is the key reason that it becomes more prone to aggregation, and when it aggregates, several non-specific interaction also happens, and thereby that is one way in which it creates the toxicity. Also it is becoming prone to proteolytic cleavage, generating the smaller fragment bearing the polyglutamine chain goes to nucleus and creates problem there also. So I have just shown very few data from this work. If you want to go into detail, this is a paper which we published in eLife in 2016. So this will have, like, almost 20 figures in that. But if you want to know more, you can refer to this paper. Okay. Then once we came to know that okay, there is a difference in structure of wild type protein and mutant protein. Next obvious question was how to take advantage of this difference in secondary tertiary structure of the mutant and wild type protein. So we immediately started to, this was the question, what next can be done based on the information we have. Okay, So next, what we did, we had the purified proteins. So we sent them to have to identify small DNA aptamers. Okay, these aptamers are small DNA sequences, and we wanted to know that if we can identify from a library of such sequences any such small sequences which can specifically bind to either wild type or mutant proteins. Okay, so what we do is we have these sequences. This is all commercially done. Okay, we did not do in our lab. What we did in our lab was to have our purified proteins labeled with Alexa flow dye. So you can see this is our purified Huntington protein labeled with an fluorescent dye, and we supply it to the company, and they have this uh, array of aptamers, and they'll add our proteins to them and see that where they bind. Okay, so they do detection, and then eventually they gave us a list of aptamers which were able to bind, sorry, which were able to bind only specifically to the mutant protein. Okay, not to the wild type protein. So now we wanted to know if these aptamers are able to specifically bind to the mutant protein. Binding of these aptamers alters what, okay, in the mutant protein. So that was the next question. So first thing we wanted to check is that because of the binding of the protein, this is one of the function of the normal wild type protein, that it is, I told you, it is working as a scaffold protein. So one of the complex which is harbored by uh, Huntington protein is called PRC2 complex. It's a polycomb repressive complex to PRC2. And what is the normal function of PRC2? It is a involved in epigenetic modification. So all of you must have heard or at least studied in your BSc or MSc that the histones are modified. They can be acetylated or methylated. ये सुना है? किसी ने सुना है?
Yes,
Yes,
sir.
sir.
Okay. And the level of acetylation or methylation determines whether it will be active chromatin, that is euchromatin, or heterochromatin. Okay. So that level of acetylation or methylation will decide the gene activity. So PRC2 as such, what the enzyme activity of PRC2, that it is methylating the histone S3 at lysine 27. Okay, and trimethylation of histone H3 at lysine 27 leads to epigenetic silencing. Okay, so basically it is an epigenetic silencer. Okay, and it interacts with Huntington. So you can see here, this is our previous data, that you have input of Huntington, you have PRC2, and when we pull down using anti-Huntington antibody, you see that PRC2 subunit, these are the subunits. EZH2 and SUSI12 are the subunits of PRC2. PRC2 has 4 subunits: EZH2, SUSI12, RBAP48, and ED. Okay, these are four. It's a multi-subunit complex. So you can see that when we pull down Huntington, the PRC2 subunits, EZH2 and SUSI12 also are pulled down. And when we did a PRC2 assay to check the level of histone S3 trimethylation at lysine 27, we can see that when we do the assay using the methyl donor, so you can see here the addition of methyl group. Okay, so more with wild type protein, you have this much methylation signal, but when you have mutant protein added, the methylation signal increases. So what it infer that in presence of mutant Huntington, there is more methylation of histone S3 lysine 27. That means if there is more methylation, there is more silencing of gene. Is it making sense? Methylation बढ़ेगा तो gene silencing बढ़ेगा. Is it making sense?
Yes, sir.
So
Yes,
what
sir. Yes, sir.
basically mutant Huntington is doing, it is increasing the methyl transferase activity of PRC2 and thereby abnormally silencing the genes. Okay, so that is what is happening. So we wanted to first check whether this particular activity of mutant Huntington is altered by our mutant Huntington specific aptamer or not. Okay, so that was one of the essay. There were several things tried, but today because of lack of time I'll not be able to show everything. So I'm just showing this, that when we added this is our aptamers, which are MS, we named them MS1 to MS4. MS is for mutant specific. So we tried 4 aptamers and we added those aptamers along with the mutant Huntington in the PRC2 assay. And as you can see, in absence of aptamer or the aptamer when they are present in 1:1 ratio, the mutant activity remained almost same except for MS3, that in 1:1 ratio also it could significantly reduce the methyl transferase activity. But at 1:10 ratio, okay, at 1:10 ratio, all four mutant specific aptamer could significantly reduce the histone methyl transferase activity of PRC2. You can see signal here also. So that means that mutant Huntington is able to restore the PRC2 activity to wild type levels, and that is in that way it is able to bring back the or you can rescue the gene dysregulation or gene transcription dysregulation. Also here also we saw that whether it is specific or not. So we did a pull down assay. So in presence of different aptamers, so you can see that when we had aptamers, so the interaction, you see that PCH2 in presence of Huntington, this is interaction, but when we put the aptamer, the interaction with the PRC2 is decreasing. Okay, so when the interaction is decreasing, that reduces the PRC2 activity also. Okay, so this also is already a published. This is a published paper. It is published in Molecular Therapy Nucleic Acids. It came in 118. So this also, if you want to go in detail, you can refer to the paper. But now we have several other papers, but I wanted to just because of the lack of time, I showed only two of them. Now we'll quickly come to what we are doing currently. Okay, so these are ongoing. इस सब लोग को समझ में आ रहा है अभी तक? Are you with everybody?
Yes, sir.
Okay.
Yes, sir.
So quickly, we are going to discuss now what we are doing currently in our lab. So we have four different projects on Huntington disease ongoing in the lab. First is we are trying to target the, as I said, PTM, post-translational modification. So Huntington phosphorylation as a potential therapeutic target. Second project is dealing with small molecules. We are using phytocomponents, curcumin and quercetin along with the MS3 aptamer, which I just showed you. So it's a combinatorial therapy to alleviate the mutant Huntington toxicity. Third is to use dietary fatty acids, which is medium chain fatty acids, which can also be used as potential therapeutics for HD. And finally, the fourth study we are trying to see that what is there in the secretome of Huntington mutant Huntington expressing cells and how it can induce the aspartogenesis and how that can be targeted. Okay? So these four projects are ongoing in the lab. One is a SERB, DST SERB supported project. Two of them are ICMR supported projects. Okay. Yeah, somebody is asking. Any question? Anybody has
नहीं
any?
कोई क्वेश्चन
Okay, so let's move forward. Okay, so what all we look in our lab? So we are using a cell line based model to address our whole studies. So we have a mutant Huntington expressing cell lines. We have a cell line which expresses full length mutant Huntington and wild type Huntington, as well as we have a cell line in which we express only the exon 1, which bears the expanded CAG repeat. So that's the truncated Huntington model. So both cell line models we use in our lab, and these are the common pathways we explore, which are already seen as in the previous slide. That we check the mutant Huntington aggregation, we check the endoplasmic reticulum stress levels, we check the mitochondrial dysfunction, we check the gene expression dysregulation, we check oxidative stress levels, we check cell viability, and we check immune parameters. Okay, so these are the different parameters we are testing, and I'll show you. So we cannot, I cannot show all four projects. There is lack of time. So I'll show you examples from one of the projects. That is the first project where
क्विकली जो है एक प्रोजेक्ट दिखा दीजिए बाकी दे विल कनेक्ट विथ यू इफ दे आर इंटरेस्टेड है ना
yes.
in
So I
one
have.
hour they will not learn everything of 20 years of an experience.
Yeah, so that's why I am just showing data from one of the project only, but you will get an idea that what kind of experiments we carry out. Okay, so I'll be focusing today on the first project, that is Huntington phosphorylation as potential therapeutic target. So here in this project, proteins, if you are able to bring back the phosphorylation status to normal, the aggregates are reduced. We are able to reduce the the ATP levels which are reduced are brought back to normal. The ROS which is increased is reduced, brought back to normal. ER stress is also reduced. So this is kind of summary of the first project which we are about to publish soon. Okay. So with this, I told you the funding agencies which have funded our work is DST SERB and ICMR. This is my team. So the work which I showed you today is mainly done by my research scholar Raju Bhai Davi, standing here. But this is my team comprising of other research scholars and my dissertation students. With this, I come to end. Thank you.